This is Ethan Norbash with the DNA replication video. What is a double helix, and describe the shape? A double helix, and in this example, is two strands these two strands of DNA. But in a real life DNA, it's intertwined, which it couldn't be in this video, but in real life in the nucleus it is. And which base bonds to adenine? Adenine, which is represented by the A, bonds to thymine, which is represented by this T. And which base bonds to guanine? Guanine, which is represented by this G, bonds to cytosine, which is represented by this C. And how do the two strands of DNA stay together? They stay together through these hydrogen bonds. And these hydrogen bonds are weak, so they can be unzipped to do the DNA replication. And what are the components of a nucleotide? A nucleotide is one phosphate, one deoxyribose sugar, and one nitrogen base. And where's the DNA found in the cell? It is found within the nucleus. And what is the end result of DNA? Oh, where in the cell does DNA do replication? It takes place inside the nucleus, because the result is also going to need to be contained inside the nucleus, and DNA is not allowed to leave the nucleus. What is the end result of DNA replication? The end result is two strands of DNA, each composed of one old strand and one new. And how are mistakes or mutations avoided? The mistakes or mutations avoided are when the helicase unzips the nitrogen bases, and then the phosphate comes and copies it over to its next to a new strand, as represented by here. It has to make sure that it copies over correctly for every single strand or doesn't miss any, and that would be the best way to avoid mutation. And what is meant by semi-conservative? Semi-conservative means that if it was that only conserves half or saves half, where only half is used and the other half is still used, but it's used in the other strand. Whereas conservative would just be the two strands are left together. And how do DNA unzipping enzymes work? How do DNA unzipping enzymes work? They work where as I've shown before, the helicase will unzip, as represented by the cotton ball with the H on it, and the phosphate will come along and it will add the new nitrogen bases and basically the whole nucleotide to the original strand, and then rebond it with the hydrogen bonds, so it becomes a full new strand of DNA. And then, what are the two roles of? What are hydrogen bonds? Hydrogen bonds, as shown before, are basically what hold these two DNA strands together, and without them, they wouldn't be able to be combined and stuck together. But they're also weak enough so that when separated, they will break apart easy enough in order to be copied, as shown before. And what are the two roles of DNA polymerase? The two roles of DNA polymerase is one, to create the new nucleotide, and two, to add it onto the original strand. We'll now fully demonstrate the DNA replication process. First comes around the DNA helicase, which will unzip and take off these hydrogen bonds from the original two old DNA strands, and then when they're separated, the polymerase comes along, and then it'll match the correct one to the matching nitrogen base, which would create this new strand. But it also adds these hydrogen bases, add it back so that this forms one strand of DNA again. and then the same process is done to the other old strand and replaced with its new strand, which is an exact copy of the other old strand, which was already bonded, and it would be like added back on through the DNA polymerase. I will now point out all the parts used in this video. So first is adenine, which is a nitrogen base, which bonds to thymine, and guanine, which will bond to cytosine. And then with that, there's another part of the nucleotide is the deoxyribose sugar and the phosphate. And the phosphate group. This whole thing right here is the phosphate group, and then the nitrogen base is any one of these: adenine, thymine, cytosine, or guanine. And the hydrogen bonds are these things that bond the two strands together, and the DNA nucleotide is the whole strand, the phosphate, the deoxyribose sugar, and the nitrogen base, and the DNA helicase is the thing which is used to unzip the hydrogen. I mean the DNA through the hydrogen bond, and the original DNA strands, which is this, and which are the two old strands, which are used to make the newly made DNA strands, which don't consist of two new strands, but one old and one new, like this. It consists of one old and one new, and that is it for the parts used in this video.